First of all, I would like to thank the organizer for inviting me to this meeting, which I was asked to give a presentation on genetically engineered models for GBM and novel treatments. I apologize also a little bit for my voice. I had a bad cold last week, and there's still sometimes a frog in my throat left, so I may take a sip every now and then. Moving forward. So actually, already some years ago, we wrote this review about mouse models, not only genetically engineered mouse models, but mouse models in general, for the purpose of preclinical testing. So when preparing for this presentation, I reread that, of course, and I found, oh yeah, our main conclusions at that time, if it comes to GEM models, was that they do recapitulate many of the characteristics of human GBM. They have been very informative for explaining some of the biological questions. However, at that time, they were not really used for therapy testing. And so how's the situation now? That we're in 2023, is that indeed, yes, GEM studies are being used for therapy intervention studies. They have been reported, but even still not that many. I will give some of those, I will give examples, also review the models and give some of the opportunities and also, of course, some of the limitations, as already correctly stated by the first speaker. Indeed, there is not a universal model that can be used for all types of studies, intervention studies, so you need multiple models. Let's go back a little bit and what are actually the hurdles in treatment of GBM. So, of course, we are now looking to this these, let's say, depressing survival curves for more than 20 years, and actually we haven't done that well in that sense. And yeah, what are the hurdles? So, of course, we spoke about the fact that it's a very invasive tumor, so a complete surgical resection is impossible. We now know that there are multiple deranged molecular pathways, alterations, which gives a high amount of inter and intratumoral an immune suppressive tumor phenotype and the microenvironment, and this is a bit my work. It's on the blood-brain barrier, which is an impediment for the delivery of drugs to the brain, and especially the role that these transport proteins play in that sense. So about already in the end of 2010, we published this article. This is about the development of a transgenic model that needed to be robust and rapid in order to use it for intervention studies. But at that time, I think the early days of the 2000s were actually quite exciting times for GBM research. And I would like to go back to two findings, and that is, of course, the finding that we were dealing with a hierarchical model where there is a so-called cancer stem cell, cancer initiating cell, next to the more differentiated cells. And in line with that, we also learned how to culture those cells better than we did in the past with serum culture medium. We changed to the so-called neural-basal medium, which allowed these cells to maintain their primitive, more undifferentiated state, rather than the differentiated models that you get when you're using serum in your cultures. And also, we were actually still in the pre-omics time of GBM classification, where we were still breaking them from 1 to 4, with 4 being glioblastoma. And then, indeed, if you looked at the hallmarks, you would get that you see the invasion, you see the microvascular proliferation, and you see the pseudopallidating necrosis. So all items of what are the GBM. And this was from a patient that was actually diagnosed for GBM after a post-mortem. So there's no surgery has been done on this patient. So you can really see all these structures that are present in GBM. Of course, we are now later, 2023. So in 2007, the Cancer Genome Atlas pilot project started, including GBM, and this learned us much more about the molecular pathways that are involved. And of course, these are important when you want to model cancer in mouse models. This was later refined, and also so we now segregate between the four different subtypes: classical, mesenchymal, proneural, and neural subtypes of cancer of GBM. So how was this modeled? This is one of the not the first model, but one of the earliest model what they actually did. And what is being done here is, so this is a germline mouse model. This means that all cells in the mouse will have this transgene, and this transgene contains of a GFAP promoter. So only cells that will express GFAP will also express this transgene. It is because it is in all cells, and because it has this GFAP, it will trigger all GFAP containing cells. And there's also a LAC-C promoter, a LAC-C reporter here, and you can see in this whole mount slide of this young mice that all the cells are expressing this transgene. Well, the transgene also has this GFAP, this RAS, this mutant RAS, and they made them in different flavors. So there is the D7 line, which has a high expression of the RAS. There was the D8 that had the lower expression, and the D8 was even segregated in a hemizygous and a homozygous. And what it basically boils back to is that if you look at the high expression, you see they develop tumors within 10 to 14 days. These animals are dead. That's not really handy to if you want to do any studies on them. So if you look at the homozygous mice, you will see that that's here. You see they also die within 4 weeks. But with the hemizygous, there is quite a reasonable latency time. But most of these tumors will be grade 2 of time. Interesting though that although you start with just GFAP only, they do lose the P16, P19, and sometimes also P10. So these are typically losses that are also seen in GBM. Then this was another model that was described, which is based only on. It was actually the first model that used only used the loss of tumor suppressors. So as you can see here in this slide, you see it loses NF1, which is an inhibitor of RAS, and it also here loses P53. And these tumors also develop, but you can see these tumors take roughly 6 to 8 months to develop. Also not really practical if you want to do therapy in intervention studies. You need to look at these mice for 6 months, and probably they have once you notice that they have a tumor, you're already too late to do any intervention. This is something similar here, but it's two different tumor suppressors. But here also in combination with so this is in particular in cells that are expressing GFAP. So these will be switched to lose P53 and P10. So not all cells in the body lose this, only cells that have expressed GFAP and P10. And also here you can see that the latency is quite long. These have been very interesting models for biology, but also not very practical for the therapy intervention studies. And the same is true for the NF1 and P53 in the neural stem cell. The nice thing that they show here with this work is actually that you needed neural stem cells in order to create tumors from these, to create GBM. And that is because they had a germline nestin promoter, and under and that drives CRE-ERT. Now CRE is able to go to the nucleus and do the CRE-LOX conversion. But if it has an ERT, that can only do this when you expose the animal also to tamoxifen. So you can really time the switching of the genes. And they did this in very young mice, and then you can see if you do it even in embryos, so you treat the mother with tamoxifen, that these tumors will develop within several weeks. But if you do that at a later time, it will take much more time for them to develop. And they also found that if you would do that by somatic switching, so you take a virus, which will give Cre, you only get tumors when you inject the Cre nearby the subventricular zone. And that is the zone where more stem cell-like neurons are present. But the latency here also is 10 to 14 months. Not really very practical if it comes to intervention studies. So this is actually another model that is being used in the literature very often. It's a so-called ARCAS-TVA model. And this is briefly how it works. So you need virus-producing cells. And these viruses, they are in chicken, they are grown in chicken cells. They can contain, they need to contain a plasmid, and the plasmid can carry your gene of interest. In this case, it's a PDGFB, but it can be all kinds of genes. And these are then introduced into the mouse. But the TVA is, the ARCAS is a virus which cannot enter into normal mammalian cells. So you need to have a mouse model that is transgenic for the TVA receptor. And they have developed TVA receptor mice under the GFAP and under the nestin promoter. So then you can more or less address which cells will be infected by this virus. And if you introduce that, as depicted here, the non-transduced cells will not be taken up the virus, but the nestin positive cells will be transformed. So yeah, Eric Holland pioneered this model, and he did quite a lot of studies with this. And here, this is just one example where he showed the combination, the collaboration between the AKT overexpression and RAS overexpression. And only in the case that you overexpress them both, you will get tumors. And only in the NTVA mice, so not in the GFAP-driven mice, and only 7 out of 27. So it's not a really reproducible model, and it has quite a delay onset. But it does have striking features of GBM, but again, not really practical for intervention studies. This is the ARCAS model, and that is they were able to convert the ARCAS model in a more useful model. And the reason how they did this, indeed, in this case, they used this PDGF-beta as a driving factor. And they used it in combination with mice that also lacked some tumor suppressors, in particular P16 and P19, but also P53. And they can also use the knockout of P10. And then if you introduce these, you can see that you get very nice survival curves at a reasonable amount of time. So this is the time in days. So you're talking about tumor development within 30 days after the injection of the virus. And also most of these were high-grade tumors. So this is a model that is being used now relatively often for preclinical studies. The group of Orland Becker thought, well, we can also use, so if we can actually, they showed that they can infect any type of neuron, so you don't need to be in the vicinity of the subventricular zone. So they injected this also in the pons of the mice with the idea to create a model of DIP-G, of midline glioma. And this worked very nicely. The only thing was, and that is a bit like, what is the limitation of this model? If you look at this contrast enhancing MRI, you see that there is quite some leakiness of the contrast into these tumor lesions. And that is not really typical for DIP-G. And the reason why this happens is that the driver is PDGFB. And PDGFB is, of course, not a normal driver for glioma, whatever. And our parasites are quite rich in the PDGF-beta receptor. So the tumor cell is creating a lot of growth factor for this. Somebody else, sorry. Never mind. So the PDGFR, so there's a lot of ligand for the PDGFR receptor, and you basically probably make these parasites, you make them crazy, and that is why you lose some of the barrier capacity of the blood-brain barrier, because this is the blood-brain barrier. This is a CE, a contrast-enhanced MRI of a DMT patient, and you really cannot see any enhancement here. And that is something that you need to take into account with all these models. They may not be so suitable for small molecule drugs, and also for antibodies, because there is a relatively good accessibility of the brain because of the fact in this ARCAS model, because of the fact that you open up the blood-brain barrier. And this is a bit exemplified here. So they used palbociclib as an agent, as an experimental agent in DIPG-driven PDGF-driven DIPG. And you can see that they do have, it's not a wonderful response, but they do find some response. But actually, from our work, we've shown that palbociclib will not enter into the brain, especially not when the transporters are absent. So these are these drug transporters that we are studying. And you see that in the absence of both, you get really good levels of palbociclib into the brain. It actually likes to go to the brain. The brain-to-plasma ratio goes up to 10 or 15, which is huge, but not in animals that have these that are proficient for these ABC transporters. Then the blood-brain barrier entry is much less. So once you have created a hole in the blood-brain barrier, you will get a lot of palbociclib into your tumor, but probably not in the tumors of these children. What these models are very useful for is for looking at immunology. Because what is shown here, and this is a very recent paper from my colleague from the NKI, Laura Akari, and she is Laila Akari, and she is studying the tumor microenvironment. And what it shows here is that if you look at the tumor mutational burden in a patient, that is quite low, but it's quite comparable to the two models that they use. So this is an ARCAS model, either INFO-AR of p53 knockout. And if you compare that, for example, with GL261, which is basically a cell line model that is very often used for immunology studies, you see the tumor is 100-fold higher here. So it also contains many more infiltrating CD3 T cells, whereas these models are very low in CD3 T cells, in line with what we find in humans. So they used it to show in this paper, and I recommend you to read it because it's really nice, is that they used this in an intervention model showing why the concurrent use of immune checkpoint blockade works less than if you do it in an adjuvant setting. It's too much detail here. It will be a separate presentation, so I won't go into detail too much here. But again, I would like to recommend you to read it. And it's just another nice example how these models can be used for intervention studies. Our own models are based on conditional LOX-P mice. So how does this work? In general, you have you make the mouse that it has a transgene where you put LOX-P sites behind your gene of interest, and that is in this case you have a tumor suppressor mouse, or if you want to drive a new gene, you will have a promoter, a stop codon that is between two LOX-P sites and your gene of interest. So once you get expression of CRE in those cells, you will get a recombination, and either you lose your knockout or your gene of interest, or you have a knockout for your tumor suppressor, or you get expression of your gene. And of course, both things can happen in the same cell, so you can make nice combinations of that. And we have used that, and I don't make these models in the sense that I'm not a molecular biologist, but these have been made in our institute. But I know how to cross mice, and so we've crossed different strains of mice in order to get the correct phenotype for modeling GBM. With the only point that at that time we used KRAS, and KRAS, mutant KRAS, is not really a thing that happens a lot in GBM. KRAS is usually it's overactivated, but the driving factors are upstream of RAS. But nevertheless, we use this as a model, and we both use GFAP-CRE as a virus as well as CMV-CRE. So GFAP should, in principle, only be expressed once you inject it in the mouse, only if it's transformed, only transformed GFP-containing cells. CMV will transform all cells. And then you get, after a few days after injection, you already see the formation of a tumor. We have used a luciferase marker for studying bioluminescence. So this is something about, a few MRIs here. What is important is that we are looking at the blood-brain barrier. So if you can see from this model, this is the model that we are discussing here. So you see it also has some opening, but it's not as leaky as, let's say, the old models like the U87 that is frequently used. And this is actually, and I will discuss this, this is a similar model, but then driven by EGFR-V3, so not KRAS. And you can also see that here there is a very modest to no opening of the blood-brain barrier. We have used this to look into to do an intervention study with CSTK464, which is a PI3 kinase inhibitor. As you can see from this plot here, we are looking at what is the role of the transporters, and we can see that if we have mice that are wild type or they are knockout for both transporters, we see that that has hardly any effect on the brain penetration. So this is a substrate drug, which is a drug which is not a substrate for these transporters, which makes it, let's say, a more or less ideal drug for GBM treatment. We also see that it has the target inhibition takes place both in wild type mice with this drug as well as in the knockout mice. So in line with the fact that we get good exposure in the brain of this compound. Now, and then we looked into the efficacy, and the idea here was, actually I should have removed these two because it's look at the green curve and the red curve. The difference in between these two curves is that the fact that these mice have either a loss of P10, which is the green one, or not, the red one. And you can see that this loss of P10 makes that the tumor development goes more quickly and the animal dies. So the survival curves are different. So our idea was that if we are going to use a PI3 kinase inhibitor, we should be able to, and we are going to use this in this model that is only driven by this P16 loss. We should be able to, sorry, we need to rephrase. So we are looking at mice that only have P16 loss to mice that have P16 and P10 loss. And the idea was that if we're going to treat these mice that also have P10 with a PI3 kinase inhibitor, that we should be able to, if we are successful, to move the survival curve from the green one to the direction of the red one. And to some extent we were able to do so, and that is shown here. But as you can see for the plots, actually we had more or less only a delay in the formation of the tumor, but once the tumor was growing, it would grow at roughly the same speed. It shows that, I think, going back to one of the first slides, is that it's these are multiple pathways that are affected, and that's also shown from this slide where we did immunohistochemistry on the treated animals, and you can see that we are able to reduce the amount of phospho-AKT, so that is the PI3 kinase inhibition effect. But we were not able to do anything with the other deranged pathways. And I think what we actually learned from this is that we need to go to clever combinations of agents, keeping in mind that, of course, that all these agents need to be able to pass the blood-brain barrier, which is quite a challenge in itself. So coming to conclusions, again, GEMs can be used. The internet is still unstable. GEMs used for efficacy studies. So the intervention, they need to be robust. They need to have a high penetrance. They need to have a reasonable latency time, and preferably, and that is using luciferase reporter for non-invasive monitoring of tumor growth. Otherwise, you're bound to using MRI, and that is, of course, much more laborious. I think the CRE-LOX-based models qualify for that, and also the ARCAS PDGF-B driven models qualify for that. Albeit that I think that the PDGF-B keep in mind that there are these effects on the blood-brain barrier. And of course, again, always, as always, the suitability depends on the research question. So the ARCAS models are probably very good models for studying the tumor microenvironment and the role of immune checkpoint inhibitors. But for small molecule drugs, probably I would prefer using different models. So with that, I would like to thank you for your attention and thank the people in the lab that have worked on this. And yeah, take any questions. But I think we're going to the session for that. Okay.